So our next presenting company is NeoStem. NeoStem is a developer of novel cellular therapies, a conglomerate, if you will, of several businesses or business models, including contract manufacturing, stem cell banking, and therapeutics. So when you think of NeoStem, think of a budding Enron, not the toga-throwing party Kozlowski, but a smaller version that brings together several business models for a common goal. Presenting for the company is Jonathan Sackner-Bernstein, VP of Clinical Development and Medical Affairs.
Thanks so much. By the early 1970s, the computer industry was really buzzing. It was taking off. We were starting to see a major transition from the mainframe computers to the mini computers. That was the beginning of a great inflection point and development of multiple industries from which we're all benefiting today. At the same time, the field of medical imaging was one where doctors were still looking at shadows, trying to interpret. I have control of these slides. It's okay, thank you. Trying to interpret the shadows, the marks, the streaks, using their imagination, relying on intuition. And a great thing happened. At that moment in the early 70s, people saw a path and realized that by applying microcomputers and mini-computers to the field of medical imaging, they could transform the field from trying to imagine what was there to seeing what was there. Now, at that point in the 1970s, the field of anatomy was very mature. We knew what the structures were. Of course, as patients, we'd prefer somebody to image them rather than open us up to take a look inside. And this transformation was very instructional. Yet at that time, even into the early 1980s, the system kept looking, applying its models, and whether it was the financial folks, the pure scientists, the medical field, they couldn't seem to figure out how to fit in this transformative product, this transformative approach, into a standard model. That was a lesson that took a long time to learn. Over time, this battle, this problem, this challenge was faced over and again. The CT struggle that took 20 years, that I know that Les smiling here remembers probably when he was a young attending trying to get someone a CT scan that needed it. It was impossible. Someone came in with a potential appendicitis, the surgeon was more likely to open them up than were you to get a CT scan to look and see whether it was warranted. Magnetic resonance imaging came along, and there was a shift in the system. People started to understand that you can't put everything new, everything innovative into your standard models. You needed to be open-minded. ICDs came. It moved faster. Monoclonal antibodies came faster. And right now, I'm excited to be here at a part of a conference where we all realize that those transitions, those adoptions of new models will actually happen faster. Because today, when you talk about stem cell therapy, there's a parallel to the anatomy as a mature science. It's called logistics. We know how to manage an airplane reservation. We know how to ship a product. It's gotten so advanced that a mom and pop shop can go onto eBay and use standard tools to create a growing business because logistics are known, they're straightforward, and in fact, we at NeoStem have figured out how to bring them to a broad array of applications within the cell therapy industry. And at the same time, we have this new science brought from the basic lab, brought from the biologists into the current model. And as we look at cell therapy, what we have is the same opportunity of taking what's known, what's perfected, the logistics that exist, that expertise, and bringing the science on top of that, interdigitating them, and ending up with products that impact patients. So I'd like to just first refer to a couple of points about PCT. Progenitor cell therapy has been around for 15 years and has taken the lessons of logistics from a broad array of industries and brought them to bear on manufacturing cell therapies. They know how to do it. They've interacted with the FDA. They've got a 15-year track record. They've got a bunch of customers, many of whom represented here, who know that logistics in the cell therapy field can be mastered. So as you look at NeoStem, let's not get caught up on how things work in terms of getting the cells from the patient to the manufacturing back to the institution where they're treating. It's logistics. We know how to do that. We don't have to worry about that. What we do have to worry about is patients who have an ST segment elevation myocardial infarction. Why? Because their natural history has not changed over the last couple decades. And these are data from Kaiser, a group that can look long-term, as you heard earlier today at lunch, with an average patient follow-up of 18 years, put in systems for the long haul and still can't impact the natural history of these patients. And if you look at the pathophysiology, you'll see all sorts of complicated slides. You can look at this and focus just on the yellow boxes. Those are the 3 targets we know need to be addressed in order to alter the natural history. And we're pretty darn good with stents following on the backbone of thrombolytics. We're pretty good at dealing with ventricular remodeling with a host of small molecules, but we really stink at dealing with infarct expansion. And that's a key driver. You have the initial infarction within the first hours, and then over several weeks that infarct expands, and it turns into the patient that Astrum is looking for. So if you bypass NeoStem, I guess that helps Astrum recruit. What happens is shown diagrammatically here, as showing an example of an apical infarct, shown in purple with a white rim around it of tissue at risk of dying in the following weeks. If you do nothing except standard therapy, nothing except standard approaches, that infarct enlarges and the heart starts to become shaped more like a basketball. We believe that by using AMR-001, a CD34 CXCR4 positive cell population, autologous, so it's essentially a personalized medicine approach to treating myocardial infarction, that we can block that process and prevent infarct expansion, which would translate into clinical benefit. You've heard a little bit about cell therapy and the body's ability to have its own repair mechanism, and that exists, except the connection isn't made. There's been no evolutionary pressure for these angiogenic anti-apoptotic cells resident in each of our bone marrows, the CD34 cells, to bother moving or responding to an SDF signal that's present in the heart, the brain, any ischemic organ. Because we're done procreating by the time we have infarcts. We're in our 50s and 60s, 70s, and 80s. And so this has not been selected, but that doesn't mean it's not there ready to do its job. And in fact, there's a lot of data that I don't have time to review today that I'm happy to share with you offline that say that CD34 cells can sprout blood vessels when you administer them through the infarct-related artery, as shown here diagrammatically. We've got a very broad and broadening intellectual property portfolio, both on composition of matter and methods, and we can review this later for those interested. But what I'd like to do is take you on a quick trip through what we've seen in clinical trials and where we're headed. In the phase 1 trial, what we were able to demonstrate was a dose-related effect, a dose-related benefit on improved myocardial perfusion measured by nuclear scans, as well as an improved preservation of myocardial tissue. And we did that by doing CMR and measuring infarct size. And we also were able to demonstrate a threshold dose of 10 million CD34 cells. Now let me just make sure that that threshold dose is emphasized in the context of prior trials. Look at the literature. There's a lot of publications over the last decade and longer about CD34 cells that all look like there might be a signal, look like there might be a benefit, but it's not quite there. Look closely at that literature, and what you'll see is that the doses employed are low, typically under 5 million CD34 cells. The literature shows, as I'll demonstrate in one slide later, that there is a dose-related effect. So identifying this dose threshold, even to an extent that satisfied the FDA, is a very important differentiator of our drug-like development program. Here's a slide that shows the data graphically, as well as here, reduced infarct size. And these are part of the data that convinced the FDA that we had, in fact, demonstrated a dose threshold effect. On the left side, you see the frequency with which patients had a drop in ejection fraction over the 6 months following therapy. And at the right, you see the incidence of a drop in ejection fraction over 6 months of therapy, and those patients treated with a minimum of 10 million cells. So no one had a drop in ejection fraction. Now, it's a phase 1 study, 15 total treatment patients, but this, with the rest of the data, were looked at in the context of the literature to convince the FDA that, in fact, 10 million cells was a threshold dose that had been established and could be used as a single dose in phase 2. Our phase 2 trial is ongoing with over 50 sites. We've enrolled over half the study. We'll have 160 patients treated in this trial. And as requested by the FDA, the primary endpoint is a physiologic one of looking to demonstrate that we are impacting myocardial perfusion along with other imaging endpoints as well as collecting cardiac adverse events. So the primary endpoint is at 6 months, and we will follow these patients in a blinded fashion out to 3 years. We will be able to report the data after the last patient has completed his or her 6-month imaging assessment. So in the next couple of minutes, what I want to do is just give you a little bit of a sense of how NeoStem views the path forward and why we believe this is such an exciting opportunity. Those of you who are focused on cell therapy have, in all likelihood, invested and followed many small molecule companies, many biologics companies, and this is also true for medical device companies. But let's focus on drugs, and by that I mean small molecules and biologics. And if you ask yourself why they fail, there are 2 very common reasons. One is that they're not as potent as one expected, and the other is that there's off-target toxicity. Simplistic way of looking at the world, but we don't have to get complicated when we're talking about how to understand risk of a drug development program. So the first is dose. I mentioned this before. The literature includes not only a lot of studies that demonstrate a physiologic impact, a very important one in terms of improving ventricular function and trending towards reduction in MACE endpoints, revascularizations, etc., but also establishes that there is a dose-related effect. And this is one example from a meta-analysis by Zimmet and colleagues that appeared almost a little over a year ago, or about a year ago, where they looked at the relationship between change in ventricular function, and remember, that's the driver of this disease history, of this disease course, as well as the relationship of CD34 count on ejection fraction. So on the panel on the left, you see there's no relationship between bone marrow-derived cells, and this is for autologous bone marrow-derived cells administered via the coronary artery, which is how we administer them. But if you look at the CD34 relationship, you see there is a significant relationship, highly significant, and this has been shown by several analyses reported in the literature. So if you want to project what the impact is of this therapy, you start out by looking at the evidence that's available. Most of us, whether we realize it or not, are Bayesians, so we look at prior information to inform our current and future decision-making. If one were to do that and look in the literature, I think you would find that this is a very important finding in any drug development paradigm. Certainly, it's an important one to the FDA that's issued 2 guidance documents on the importance of establishing dose-related effects. The second thing that you look at in a drug development program is to understand the off-target toxicity risk. Now, that's really hard to predict. After all, let's think about it for a second. This is a cell therapy. How are we going to know what it's going to do, where it's going to go? Well, first of all, there's a big advantage in autologous from a safety point of view to start with, because we don't have to worry about an immune reaction. And if you have a 300-patient or a 1,000-patient database with allogeneic, you've not ruled out that you're not going to run into a significant immune reaction. Let's be very clear about that from a statistical point of view and a regulatory point of view. That's really an established fact. But what's really interesting is if you look at autologous bone marrow and you look through the literature, I'm homing in myself on an exact number, but I think it's going to end up being somewhere on the order of 500,000 people have been treated with autologous bone marrow stem cell therapy delivered systemically. That's bone marrow transplant. So if you look at that and you look really hard, you actually can find one off-target toxicity, which is a flushing reaction to one of the excipients in a stored bone marrow product that we don't use. So the issue, the risk of off-target toxicity is about as close to zero as any product would ever have at its back as it starts going through the regulatory process. If you look through the literature also, you'll find several meta-analyses that give you a sense of the safety and efficacy that we're likely to see. This is one from the Cochrane Review Group, and you can see that the point estimate at the bottom is on the left side of Unity, suggesting a favorable impact on outcome. And if you look at this again using a Bayesian lens, I think what you'd conclude is that the published meta-analyses, the raw studies, all of the scientific literature that exists say this therapy is likely to work. Thank you.